Lecture number 2 for chapter 25, we're going to start talking about the process of urine formation. And this occurs in the little structures, the little blood vessels where filtration of the blood actually occurs are called glomeruli. Glomerulus is the single form of that word, and this process of filtering the blood and beginning the process of creating urine is called glomerular filtration. Okay, some more statistics here about urine formation and how important it is. The kidneys, your two kidneys together, filter the body's entire plasma, the liquid portion of your blood, 60 times per day. So again, that just illustrates for you how important it is to filter the blood and make sure that we get rid of wastes and toxins and regulate properly the levels of various electrolytes and other substances. So the filtered plasma, your early urine that's forming in the kidneys, is called the filtrate or the glomerular filtrate. And so pretty much everything that's in the blood plasma, and remember the plasma is the liquid portion of the blood, so you're excluding the cells. You're not including red blood cells, you're not including any of your white blood cells, and you're also not including the platelets. So those are big things blood-wise. And those are not supposed to be filtered out of the blood and into the urine. If you do get blood in your urine, that lets you know that you have very likely have some kidney damage going on. However, proteins in your blood, remember how important proteins are. There are many different kinds of proteins that play various functions in the body. We have many different kinds of proteins in our blood, like albumin and the immunoglobulins, antibodies that help fight infections. And those are big molecules. They're large organic molecules, and they're too big to get filtered out of the blood. So you don't generally wind up with protein in your urine. If you do have protein in your urine, that's another sign that you may have some kidney damage occurring. Okay, now look at this other statistic over here. Urine, which you actually pee out of the body, the volume of that is less than 1% of the total filtrate. Okay, so that's 100% here would be the volume of this fluid as soon as it's first created, as soon as it first gets squeezed out of these blood vessels during urine formation. The volume is down to less than 1%. So that's letting you know that a lot of this plasma, of course, contains tons of water in it, and most of that water, most of that fluid gets reabsorbed back into your blood and other body fluids. We've talked about that before, reabsorbing water. Imagine if that did not happen. You would get dehydrated very, very quickly. So the urine, again, will contain the waste and other unneeded substances. So we're getting rid of what we don't need, and we're going to try to hang on to or keep what we do need. There are three major mechanisms that occur during urine formation. And the first step is going to be this glomerular filtration. That's going to be the major subject of the first two video lectures on urine formation. Then there's a process called tubular reabsorption. So we took a look at a diagram of one of these nephrons, these little functional units in the kidneys where urine formation takes place on the last video lecture. And it was a series of tubes up in the cortex of the kidneys and also hanging down into the medulla. Reabsorption means substances are taken out of the urine and returned to your body fluids, so things that you're going to hang on to. That's one process. Okay, so tubular reabsorption is where you hang on to the good stuff. You don't pee it out. So all glucose, for example, is supposed to be reabsorbed because glucose is a nutrient. We want to hang on to that. Amino acids, also an important nutrient, an important building block for the cells in your body to use to build the proteins that they need. So we don't want to pee those out either. We need to hang on to amino acids so that our cells can make their proteins. 99% of the water, most salt, sodium chloride, sodium ions and chloride ions, Most of these we reclaim back out of the urine, and other components of the blood as well. So this process up here, glomerular filtration, all this stuff gets squeezed out. So there's no selection other than proteins not being able to move out of the blood and into the glomerular filtrate or early urine. Everything else gets squeezed out, and then you have to go through this process of reclaiming the things you want to hang on to. I know that might seem a little strange, but that's just the way it works. And finally, there's a third phase of urine formation called tubular secretion. And some of the terminology here can be a little bit confusing, so you just kind of work through it until you get used to it. This means the opposite of reabsorption. So there can be things that are in your tissue fluids and your extracellular fluids that need to be put into the urine after this step, and that's called tubular secretion. So things move out of the tissue fluids that are surrounding these nephrons in the kidneys and move into the developing urine. So that's called tubular secretion. That's something that's done selectively. One thing that can be moved into the urine in this way are hydrogen ions, which influence your pH. All right, so let's talk more about glomerular filtration and what this means. First, this is a passive mechanical process, which means it's driven by hydrostatic pressure, the fluid pressure that's within these blood vessels that are bringing the blood into these units in the kidneys. The glomerulus is really a capillary that's porous and allows things to pass across the walls of the capillary very easily in order to filter the blood. And again, you have gazillions of these scattered across, located all across the cortex of both kidneys, or the cortices of both of your kidneys. Okay, the glomerulus is very good at filtering the blood because the membrane, or the wall, is very permeable, so it lets lots of things pass across, and there's also a lot of surface area in there. So we talked during respiratory, we talked about how surface area is important for gas exchange. In digestive, we talked about how surface area is important for absorption of nutrients and fluids. And here we're talking about how surface area again plays an important role. You need lots of surface area to do lots of filtration of your blood fluids. Also, another reason the glomerulus is good at filtering is because the blood pressure in there, the hydrostatic pressure, is around 55 millimeters of mercury, and that's quite a bit higher than the typical capillaries in the body. So that higher pressure, remember, hydrostatic pressure pushes things out, so that's going to push the fluid and most of the dissolved substances in the blood out of these glomeruli and into these structures that are going to capture and contain the developing urine. All right, if you are a molecule that is greater than 5, nm stands for nanometers, and a nanometer is 1/1 billionth of a meter. A meter is a little over 3 feet. So molecules that are bigger than that are not filtered as long as these structures in the kidneys are not damaged. And so that includes the proteins, these big organic molecules that are located in the blood. All right, if you guys remember when we talked about what actually drives the exchange of fluids out of capillaries, between capillaries and surrounding tissue fluids, it was the colloid osmotic pressure. And colloid here is referring to these large molecules, these proteins that are located in the blood. So they have an osmotic pressure. They're solutes, and solutes suck water across membranes or walls of capillaries. So you're going to have, just like we saw when we were talking about how fluids move from between the blood and the interstitial tissue fluids, you got the same thing going on here. You've got a competition going on between the hydrostatic pressure inside a glomerulus and the colloid pressure, the proteins in there trying to suck fluids back in. Okay, so this is a very nice diagram from your textbook, which is showing you a glomerulus and the blood flow into these. Okay, so some things you need to know. Many of these things will be covered in more detail on the anatomy video for the urinary system. But again, here's a typical nephron, and blood comes in toward each nephron through a tiny microscopic blood vessel called an afferent arteriole. So afferent means incoming. So we've seen that before when we were covering the lymphatic system. You had afferent lymphatic vessels that were bringing fluids to the lymph nodes. Oops, that's not the color I picked. All right, so we got an afferent arteriole. So you got blood coming in, and this wad that you see here, that is the glomerulus inside there. I don't even know if that's labeled here on this diagram. This whole thing in here, that's your glomerulus. So the glomerulus is actually a blood vessel. It's a capillary that's very leaky and is very good at allowing fluid and most solutes to pass across. The glomerulus is covered by the glomerular capsule. So this yellow structure you see here, that's called the glomerular capsule. And then the space in between that I'm kind of highlighting here with the slashes, that's where your glomerular filtrate will be located. Okay, so kind of in a nutshell, what's going on is blood is incoming through the afferent arteriole, and then it passes through the glomerulus in here. And again, you've got a high hydrostatic pressure in there, or blood pressure, and that is a very leaky capillary. So fluids and solutes get pushed out. That filtered fluid ends up over here in the glomerular capsule. And that fluid is going to start its journey through the nephron, through this structure. It's going to travel in that direction through the tube that you see there. The filtered blood comes out over here through the efferent arteriole. So we've seen that word before too. Efferent vessels, structures carry fluids away from something else. So afferent arteriole is bringing blood to the glomerulus. That's where filtration of the blood takes place. And the filtered blood exits through the efferent arteriole. And another structure that we'll see, we'll be talking about in more detail, this structure here, the juxtaglomerular apparatus. Juxta means next to. You're next to or close to the glomerulus. That's where it gets its name from. And this apparatus actually consists of everything that you see here. And that apparatus, we'll mainly talk about the macula densa cells they've got highlighted there, and these granular cells that you see right in here and also over in here as well. All of those things together make up this JGA, or juxtaglomerular apparatus, and we'll see that that plays a really important role in regulating how fast blood gets filtered as it's passing through a glomerulus. And it also plays a really important role. You know, we've talked about renin before and how that's important for long-term blood pressure control. Well, the cells that actually secrete your renin are right in here. They're these granular cells that are located right where the afferent arteriole and the glomerulus are coming together. Those are the cells that actually secrete renin. So we'll be talking about those as well. All right, so here's a diagram from your textbook which is showing you the competing forces that lead to glomerular filtration. All right, so you got blood coming into a glomerulus through an afferent arteriole. And again, within the glomerulus, this is your hydrostatic pressure, or your blood pressure that you have inside that glomerulus. That's about 55 millimeters of mercury. And again, that is pushing out fluid and solutes out. The osmotic pressure that you see here of 30 millimeters per mercury, that is due to the proteins that are, it's like my, sorry, my drawings aren't really showing up here on the screen. Let's have a lag going on with my recording software here. All right, sorry about that, a little glitch here with communication between the iPad and the computer. All right, so within the glomerulus, you've got this hydrostatic pressure that is pushing fluids, solutes out. Osmotic pressure, this is the osmotic pressure within the glomerulus. So the blood that is passing through the glomerulus has a high osmotic pressure. And again, that's the colloid pressure that's due to the protein molecules that are not getting filtered out. Those proteins are attracting water back into the glomerulus. So that's sucking in or pulling in. And then finally, you do have a hydrostatic pressure of the glomerular capsule. So that's your hydrostatic pressure of the glomerular capsule, not the glomerulus, but the capsule itself. This structure that has borders, you have fluid inside there, so there is going to be some hydrostatic or fluid pressure in there as well, but it's only 15 millimeters of mercury. All right, so if you total these things together, that pressure is trying to push fluid back in to the glomerulus. So if you total those things up together, you have 55 millimeters of mercury of hydrostatic pressure pushing out, 30 millimeters of osmotic pressure pulling fluid back in, and 15 millimeters of hydrostatic pressure trying to push fluid back into the glomerulus. And when you add those things up, your net filtration pressure, or NFP, is about 10 millimeters of mercury. That means the pushing out pressure overcomes the pressures that are trying to draw fluids back into the glomerulus. And so because of that, that's what's driving fluids to move out of the blood and into these glomerular capsules. And again, I just want to emphasize here, water's coming out and most solutes. So even good stuff at this point, like glucose and amino acids, things that we're going to need to reclaim before we excrete this urine, sodium, potassium, calcium, all your important electrolytes, they're getting squeezed out. So we're going to have to reclaim those before we excrete this urine that is forming in the kidneys. Okay, so the volume and or the volume of filtrate that is created per minute when you total that up across both kidneys, that's called the glomerular filtration rate, or GFR, and that's about 120 to 125 milliliters per minute. So that's the amount of fluid you are squeezing out of your blood every minute. So you can see why it's so important. Think about, you got about 5 liters of blood total. It wouldn't take you very long to squeeze all the fluid out of your blood if you were not able to reclaim most of that fluid. The GFR is controlled by 3 different things. One, the total surface area available for filtration. You know, how many functional glomeruli do you have? And also, you know, what is their surface area? The filtration membrane permeability. So is it leaky enough to let fluids and solutes out rapidly? And then the net filtration pressure. That can be controlled, as we'll see, by allowing more blood to flow into the glomeruli or less blood to flow into the glomeruli. It's very important to control GFR. Think about if you were getting dehydrated and you still had a high GFR, glomerular filtration rate, you would become even more dehydrated. So there are very important reasons to control GFR as part of our homeostasis. All right, in the next video lecture, we are going to talk about the tubular reabsorption and secretion processes, which are the other two processes of urine formation. Then we're actually going to go back and talk about how the glomerular filtration rate is controlled, because that's absolutely critical for the homeostasis of our bodies. And so those things will be the subjects of the next few video lectures.